Hello everyone, welcome to the B.Pharm special program where we guide you to study better and in smart way. This is Swapnil Patil going to take one of the most interesting and learning topic of B.Pharm 5th semester on the important unit enzyme 2, which is interesting unit of the subject biochemistry. Are you ready? Our topic is divided into the three modules. Module 1, that is the mode and mechanism of enzyme action. Module 2, that is enzyme inhibition. Module 3, that is isozymes and their importance in diagnosis. Let's begin with Module 1. Enzymes. Enzymes may be defined as biocatalysts synthesized by living cells. They are protein in nature, exception RNA acting as a ribozymes, colloidal and thermolabile in character, and specific in their action. Mechanism of enzyme action. Catalysis is the prime function of enzymes. The nature of catalysis taking place in the biological systems is similar to that of non-biological catalysis. For any chemical reaction to occur, the reactant have to be in an activated state or transition state. Enzymes lower activation energy. This is one of the mechanism of enzyme action. The energy required by the reactant to undergo the reaction is known as activation energy, and the reactant when heated attain the activation energy. The catalyst or the enzymes in the biological system reduces the activation energy, and this causes reaction on to proceed at lower temperature. Enzymes do not alter the equilibrium constants and they only enhance the velocity of the reaction. The enzymes lowers the energy barrier of reactant, thereby making the reaction go faster. The enzymes reduces the activation energy of the reactant in such a way that all the biological systems occur at body temperature, that is below 40 degrees Celsius. While in this mechanism of action, enzyme substrate complex formation, that is very important concepts. Let's start with that concepts. The prime requisite for the enzyme catalysis is that the substrate must be combined with the enzyme at the active site to form enzyme substrate complex, which ultimately results in the product formation P. A few theories have been put forward to explain mechanism of enzyme substrate complex formation. Theories like lock and key model or Fisher's template theory. This theory was postulated by German biochemist Emil Fischer, and this is in fact the very first model to propose the explain enzyme catalyzed reaction. According to this model, the structure or conformation of the enzyme is rigid and the substrate fits to the binding site, now active site, just as key fits into the proper lock or hand into the proper glove. Thus, the active site of an enzyme is rigid and pre-shaped template where only a specific substrate can bind. Second theory related with this enzyme substrate complexation, that is induced speed theory or Coastlands model. The Coastlands in 1958 proposed a more acceptable and realistic model for the enzyme substrate complex formation. As per this model, active site is not rigid and pre-shaped. The essential features of the substrate binding sites are present at the nascent active site. The interaction of the substrate with the enzyme induces a fit or conformation change in the enzyme, resulting in the formation of strong substrate binding site. Further, due to the induced speed, the appropriate amino acids of the enzymes are repositioned to form active site and bring out the catalysis. The induced speed model has sufficient experimental evidence from the X-ray diffraction studies, allosteric modulators and competitive inhibition on enzymes. The third most important theory, that is substrate strain theory. In this model, the substrate is strained due to the induced conformation change in enzyme. It is also possible that when substrate binds to pre-formed active site, the enzyme induces a strain to the substrate. The strained substrates leads to the formation of product. This is all about enzyme and enzyme mechanism of action. Let's begin with module 2, that is enzyme inhibition. Enzyme inhibitor is defined as substance which binds with the enzyme that brings about a decrease in catalytic activity of that enzyme. The inhibitor may be organic or inorganic in nature. There are three broad categories of enzyme inhibition: reversible inhibition, irreversible inhibition, and third is allosteric inhibition. Let's start one, that is reversible inhibition. The inhibitor binds non-covalently with the enzyme and the enzyme inhibition can be reserved if the inhibitor is removed. The reversible inhibition is further subdivided into one, that is competitive inhibition, second non-competitive inhibitors. In first competitive inhibition, the inhibitor which closely resembles the real substrate is regarded as substrate analog. The inhibitor competes with the substrate and binds at the active site of enzyme but does not undergo any catalysis. As long as the competitive inhibitor holds the active site, the enzyme is not available for the substrate to bind. The relative concentration of the substrate and inhibitor and their respective affinity with the enzyme determines the degree of competitive inhibition. The inhibition could be overcome by high substrate concentration. The enzyme succinate dehydrogenase is a classical example of competitive inhibition of succinic acid as its substrate. The compound, namely malonic acid, glutaric acid, oxalic acid, have structural similarity with the succinic acid and compete with substrate for binding at the active site of SDS. A methanol is toxic to the body when it is converted to the formaldehyde by an enzyme alcohol dehydrogenase. Ethanol can compete with the methanol for ADH, that is alcohol dehydrogenase. Thus, ethanol can be used in the treatment of methanol. Let's look into the pros and cons of this. Antimetabolite. These are the chemical compounds that blocks the metabolic reactions by their inhibitory action on enzymes. Antimetabolites are usually structural analog of substrates and thus are competitive inhibitors. They are used for cancer therapy, gout, etc. The term antivitamins is used as for antimetabolites which blocks the biochemical action of vitamins causing deficiencies. Example, sulfonilamide, dicumarol. Now let's move towards second type, that is non-competitive inhibition. The inhibitor binds at a site other than active site enzyme surface. This binding impairs the enzyme function. The inhibitor has no structural resemblance with the substrate. However, there usually exists a strong affinity for inhibitor to bind at the second site. In fact, the inhibitor does not interfere with the enzyme substrate binding, but the catalysis is prevented, possibly due to the distortion in enzyme conformation. Heavy metals also lead to the formation of covalent bonds with carboxyl group and histidine, often resulting in irreversible inhibition. Second type, that is irreversible inhibition. The inhibitors binds covalently with the enzymes and inactivate them, which is iodoacetate is an irreversible inhibitor of the enzyme like papain and glyceraldehyde 3-phosphate dehydrogenase. Iodoacetate combines with sulfhydryl group at the active site of these enzymes and makes them. Diisopropyl fluorophosphates is a nerve gas developed by Ceremons during the Second World War. Diisopropyl fluorophosphate irreversibly binds with the enzymes containing serine at the active site. Example, serine proteases, acetylcholine esterases, and many organophosphorus insecticides like melathonin are toxic to the animals, including man, as they block the activity of acetylcholine esterase, essential for nerve conduction, resulting in paralysis of vital body functions. Example like disulfiram, which is known as antabuse, is a drug used in a treatment of alcoholism, which irreversibly binds with enzyme aldehyde dehydrogenase. Alcohol addicts, when treated with a disulfiram, become sick due to the accumulation of acetaldehyde, leading to the alcohol avoidance. The penicillin antibiotics acts as an irreversible inhibitor of serine containing enzymes and blocks the bacterial cell wall synthesis. Irreversible inhibitors are frequently used to identify amino acids residues at the active sites of the enzymes and also to the understand mechanism of enzyme action. Third type, that is allosteric inhibition. The details of this type of inhibition are given under the allosteric regulation as a part of regulation of enzyme activity in the living system. This is all about inhibition, enzyme inhibition. Now let's move towards module 3, that is isozymes and their importance in diagnosis. Isozyme. Isoenzymes, the multiple forms of enzymes catalyzing the same reaction as known as isoenzymes. They, however, differ in their physical and chemical property, which includes the structure, electrophoretic and immunological properties, pH optimum, relative susceptibility to inhibitors and degree of denaturation. Explanation for the existence of isoenzymes. Many possible reasons are offered to explain the presence of isoenzymes in the living system. Isoenzymes synthesize from different genes. Example, malate dehydrogenase of cytosol is a different form that found in mitochondria. Oligomeric enzymes consisting of more than one type of subunits. Example, lactate dehydrogenase and creatine phosphokinase. An enzyme may be active as monomer or oligomer. Example, glutamate dehydrogenase. In a glycoprotein enzymes, differences in a carbohydrate content may be responsible for isoenzymes. Example, alkaline phosphatase. Structure of LDH isoenzyme. LDH, lactate dehydrogenase, is an oligomeric tetrameric enzyme made up of 4 polypeptide subunits. Two types of subunits, namely M for muscle, H for heart, are produced by different genes. M subunit is a basic while H subunit is acidic, and the isoenzymes contain either one or both the subunits, giving LDH 1 to LDH. Friends, this is all about isoenzymes introduction. What are the diagnostic importance of these isoenzymes? Isoenzymes of LDH have immense value in the diagnosis of heart and liver related disorders. The healthy individuals, the activity of LDH2 is higher than that of the LDH1 in serum. In the case of myocardial infarction, the LDH1 is a much greater than LDH2, and this happens within 12 to 24 hours after the infarction. The increase in activity in serum is an indicator of liver disease, and isoenzymes activity in RBC is 80 to 100 times more than that in the serum. Hence, for estimation of LDH or its isoenzymes, the serum should be totally free from hemolysis, or else false positive results will be obtained. As many as 6 isoenzymes of alkaline phosphatase have been identified. This is all about isoenzymes and their diagnostic applications. Now friends, let's move towards conclusions. With all these information, there we come to the end of the topic. Enzyme may be defined as biocatalyst synthesized by living cells. They are protein in nature, exception that is RNA acting enzymes, which are ribozymes, colloidal and thermolabile in character and specific in their action. Catalysis is the prime function of enzymes, and the nature of catalysis taking place in the biochemical system is similar to that of the non-biological system. For any chemical reaction to occur, the reactant have to be in activated state or transition state. In all modules, we discuss mechanism of enzyme action, detailed account of enzyme inhibition and concept of isoenzymes. Lastly, we covered the details of importance of isoenzymes in diagnosis. Dear students, don't forget to review all of them in your self-study. If you have missed any of the module or content, you may log on to our website www.cec.nic.in to download videos, FAQs, LOR and other contents so that you can stay updated with this chapter. Keep studying. Good luck. Goodbye.